Hello everyone. Good evening to all of you. I hope so I am audible to everyone. Can you please confirm? And the screen is also visible to all of you? Yes? Okay. So welcome to the 8th day of CUET CUET UG Vijeta Series. As you know, we all have started this session from 28 June, which will be lasting till 13th July. And you will be having some important topics being covered in this session. So far, I guess most of you might have got greater benefits from these sessions because we are covering only those topics that are important from examination point of view. Right. So today we are moving on to Unit 3. And under Unit 3, any guesses what is the topic could be? Looking into the picture. Any guesses what we are going to discuss today? Yes, we are going to discuss today regarding cancer. Okay, so before moving into this topic, let me just remind you to join our BioTecNika in Telegram app, or you can download this app directly from Google Play or App Store. Now, what is the benefit of downloading this app? You will get all the notifications regarding exams, regarding admissions, whatever the occurrences that occurs in the field of life science, everything will be shown or will be notified to you in just one single platform, that is Biotech app. So what are you waiting for? You can easily download or just join the Telegram app. So now we are going into the session today. So before going into the session, one simple question to you. What are we made up of? We are made up of what? How we are organized? We are made up of cells, right? And you can see cells contain what? Cells contain chromosome. And chromosome is made up of what? Gene. So what is the significance of gene? What is the significance of gene? Gene is considered to be the functional and basic unit of life, right? Because it is being transmitted to our daughter cells. We are getting inheritance, right? So now why I am speaking of gene when I am taking the session for cancer? Because there is some defect or mutation that occurs in this gene which ultimately leads to cancer. So if you see, what is a cancer basically all about? Cancer is nothing where you will find uncontrolled division of cells where you don't have any control. Normally what we are having a balance. The cell will grow and divide only up to a certain limitations. Beyond that, either it will undergo processing or it will undergo death. But when it comes to cancer, what happens? We don't have such limitations and they will start growing uncontrollably. That disease is known as cancer. So, when a person is suffering from cancer, the major change that occurs in his system will be the change in the DNA. And you all know that DNA is one of the main controller of cell cycle. Cell cycle means what? These are the sequence of events which occurs during cell division, right? So this phase will prevent the cell from staying in interface for the normal period of time. Now what happens? Normally, when a cell cycle happens, we will have certain checkpoints. What is the purpose of checkpoints? Checkpoints will give you ensurety. Ensurety regarding what? Ensurety that everything is fine, the road is clear, and you can move ahead. But when it comes to cancer, the checkpoints also fail. So what does it leads to? It leads to accumulation of mutations. Meaning if there is a damage, defect, anything that is present within the DNA, it leads to the accumulation. More or less, it occurs in the somatic cells that are responsible for regulating the cell growth. So if you consider, almost every tissue in our body are prone to be cancerous. But one point to be considered over here is those cells which have the ability to undergo division and reproduction throughout the life. That means almost all cells in our body are prone to be cancerous. Right. Next, what we are having. So, what kind of genetic factors that is contributing to these kind of instability? Instability in the sense where the DNA is not there in our control. The reason could be any defect that occurs within DNA replication, as you might have heard about fundamental process. What does fundamental process indicates? Where DNA will be transferred or converted to mRNA, you can say transcribed, and mRNA will be translated to protein. So any defect in the first step can also put a cell to undergo cancer. Suppose if we have a damage within the DNA, at that time we have some repair machineries that will be recruited at the position. So definitely, if any defect occur in this repair machineries, what does it leads to? What does it leads to? The repair will not be, the damage will not be repaired. Third category is, as I said you, cell cycle checkpoints. If any checkpoints are damaged, then what happens? It will not give you the ensurety whether to go ahead or not. Next, mistakes in mitosis. And last but not the least, abnormal chromosome numbers. You might have heard about it. This is a kind of ploidy. You can say ploidy mutation where there is abnormal number of chromosomes will be there. So any of these reasons can contribute to cancer. So moving on to the genetic alterations. What are genetic alterations? Genetic alteration in the sense, what are those changes that cancer cells are going to take advantage of? Advantage of what? For undergoing growth. For example, when we look into normal cells, does normal cells have some restrictions? Yes, normal cells do have some restriction. Normal cells do have some limitations. It will not grow out of the boundary. But when you see a cancer cell, they are free from any such restrictions or limitations. Now, what could be those restrictions? That restriction could be, for example, normal cells needs an inducer to undergo cell division. So something has to occur. Cell division, cell reproduction, whatever be the case, you need an inducer or you need a stimulant that will help in undergoing, that will give instructions to undergo cell division. Next, normal cells will not survive if they have irreparable changes. Irreparable changes means those damages which cannot be repaired. In that case, normal cells will not survive. They will direct to apoptosis. Anyone having the idea what is an apoptosis is? Anyone having an idea what is an apoptosis is? Have you heard of the term apoptosis? You might have heard. It is related to cell death, right? Programmed cell death. Next, last but not the least, normal cells. Yeah, exactly, Anit. Normal cells will not wander away from the location and start making colonies. Rather, they will be restricted to a particular position, or else they must get some signal for wandering. They will not wander actually, they will remain as such. So cancer cells, you can see they are free from all these kind of restrictions. That means they do not need a stimulation. They can go irrespective of any kind of damage and they can wander and make colonies anywhere inside the body. So, if I see the three most important characteristics of a cancer cell will be, it will undergo proliferation uncontrollably. Second, it will be producing malignant tumors. Third, it will be invading healthy tissues, that is metastasis. Meaning what? It will be spreading to different parts of the body. So these are, of course, we are having lot of properties, but among them, these are the most important one. Next, if you compare a normal to a cancer cell, what could be the difference you can see? Yes. How can you see a difference between normal cell and a cancer cell? Anyone? Yeah, we'll come to that, Anit. But before, let me know what is the difference between a normal cell and a cancer cell? Anyone? See, normal cells are what? These are small, uniformly shaped nuclei. You can see there will be, nucleus will be small. So when the nucleus will be small, they will be having large cytoplasmic content. But check with cancer cells, what they are having. They will be having irregular shaped nuclei, because of which the cytoplasmic ratio is less. Right? Now looking into the size and shape. So they are having a specific structure. They are having a specific size. Right? Size and shape. That is what you call it as a conformity. And also they are arranged properly. But if you look into a cancer cell, all the cells are different in size as well as in shape. That means it is a kind of disorganized arrangement. Right? Next, what you are having? Cell surface markers. Here, even though you are having cell surface markers, but there will be normal expression or normal presentation. But in this case, you will find elevated or excess expression of cell surface markers. Last but not the least is the division of cells. When you look into the cell division status, these are slow. And also they are clearly demarcated. Demarcated means they are having specific boundaries. But when you look into the cancer cells, you will find they don't have any boundaries and they will have increase in the rate of cell division. Right? Yes, you are correct, Anas. So these are the basic differences between a normal cell to that of a cancer cell. Now, if a normal cell gets converted to a cancer cell, what are the changes that a cell will undergoing? First, it will be sustaining proliferative signaling. Meaning what? No matter what, the cell will undergo proliferation. They are independent of any kind of signals. What do you mean by proliferation? What do you mean by proliferation? Proliferation is nothing but increase in the number. Right? Second, evading the growth suppressors. Meaning what? Definitely, our body is equipped with some defense mechanisms. And one such defense mechanism is the presence of tumor suppressor genes, about which I'll come into details in the upcoming slides. So, as the name indicates, it is suppressing something, or you can say inhibiting something, or you can say stopping something, right? So, what happens? They are escaping that mechanism as well. Third, they will activate invasion and metastasis. That means they are invading different parts of the body, or they are entering into different parts of the body, and they are spreading to different locations. Fourth, enable the replicative immortality. That means continuous cell division will be there. There will be no end to that. Next, what you will be having? Angiogenesis. Any idea what is an angiogenesis is? What is angiogenesis? Any idea about that? Anyone? What is angiogenesis? Angiogenesis involves formation of what? Formation of blood vessels. Right? Next, the last point will be resistance to cell death. Yeah, formation of blood vessels. Resistance to cell death. That means no matter what, the cell will never undergo cell death. So these are some changes which you see when a cell has converted to cancer. Next, what we are having, what is the cellular basis of cancer? Cellular basis of cancer is nothing but it is related to division only. If you see, we have got a normal cell division. So we have got a cell that is undergoing division. Suppose if we have a damage, either the damage is repaired. In case if it can't be repaired, it will undergo apoptosis. So we have got two alternatives for that. But look into the case of cancer cell division. First mutation, second mutation, third mutation, and fourth mutation. So there is increase in the mutation rate. And this leads to what? Uncontrolled growth. So in a normal cell, if you see, there is a balance. Balance between what? Growth of a cell and death of a cell. So when you get into a cancer, when a person get into a cancer, this balance gets interrupted. And main interruption is because of these two reasons: uncontrolled cell growth and unable to undergo apoptosis. Next, looking into the types of tumor. So what are the two types of tumor? We have got benign tumor and we have got malignant tumor. Can anyone say among these two tumors, which is prone to be cancerous or which has the ability to undergo cancer? Anyone? Among malignant and benign, which one of them is prone to be cancerous? It will be your malignant one. Why? Because when it comes to benign tumor, you can see that there will be group of cells, but that group of cells will be present in a single mass. No doubt they will be proliferative, but present in single mass. Very correct, Anit. Malignant, you can say this is a cancer causing tumor. Why? Because it is moving to outside. Why? Because it is undergoing metastasis. Right. So that is the basic difference between benign tumor and a malignant tumor. So if you compare the two tumors, benign is non-cancerous and they are having a capsule, not invasive, growth rate is slow, they will not undergo spreading and the cells behave normally. But if you look into the malignant tumor, they are cancerous, they do not have any capsule, it will be fast growing, they can undergo metastasis. Compared to the structure, the cells will be abnormal. They will be large, dark nuclei, and they will be having no boundaries when they are growing. Now, you understood about types of tumor. Next, let's discuss about what are the types of cancer. First category is carcinoma. What is carcinoma? Any cancer that begins in the tissues like skin or that covers the internal organs. It could be adenocarcinoma. It could be basal cell carcinoma, meaning that occurs within the basal cell, commonly skin cancer only. Squamous cell carcinoma that occurs in the squamous cells. Transitional carcinoma, which usually occurs in the urinary tract, etc. Sarcoma, meaning the cancer that affects our connective and supportive tissues like bone, cartilage, fat, muscles, as well as blood vessels. Leukemia, as the name indicates, this is related to our WBCs. It usually starts in the tissues that make the blood cells, such as the bone marrow. Then lymphoma and myeloma. What does this indicate? These are affecting our immune system. So any cancer that affects our immune system is considered to be lymphoma or myeloma. And if it is affecting our central nervous system, you call it as brain and spinal cord cancers. So these are the general classification of cancer. Other than that, we have some other terms like germ cell tumors, which where the tumor occurs within the cells that give rise to germ cells, like sperm or eggs, or the tumors that arises into endocrine cells, like the cells responsible for releasing hormones, like we are having thyroid, pancreatic, anything. Carcinoid tumors. Carcinoid tumor is an example or a type of neuroendocrine tumor, but they are exclusively found in GI system, gastrointestinal system, and mainly affecting what? Rectum and small intestine. Okay. So these are some overall idea about different types of cancer and tumor. Now, we are going to discuss about all these properties in details, like how they are related to cancer cells. Okay, and this is definitely going to be an important topic when you go into the cancer as a subject. So, the first one is loss of growth control. What happens in this case? We are growing normal cells and we are growing tumor cells. In this case, what happens? Normal cells will undergo proliferation, and up to a certain stage they will proliferate, they will become quiescent. So basically, what they are forming? They are forming a monolayer, single layer. But see tumor cells. If we are growing tumor cells in a Petri plate, you will find cell proliferation is continuing. So this is not a monolayer. Rather, this is a multi-layered clumps. The first important feature. Second important feature, we are having the cells are immortal. As I said to you already, normal cells will have a limited capacity for cell division. So after reaching a finite number of divisions, they will undergo the process of aging. But when you look into the cancer cells, they will never undergo aging. Why? The main reason is that they have got an enzyme known as telomerase, which helps in remaining active. And coincidentally, this telomerase enzyme is not present in normal cells. And that is what related to aging process in case of us. So because of this, they divide continuously. Third, errors in DNA. As I said you already, when you look into the normal cell, DNA replication will be slower, right? But when you look into the cancer cells, it will be faster. And in that faster rate, they will have higher aberrations. Aberrations in the sense mutations. Moreover, the checkpoints will also be having some defect. Okay, as I said you about the cell cycle checkpoints. So normal cell induces apoptosis when the genetic content becomes highly mutated. But cancerous cell, they will escape this apoptotic pathway. They will never undergo apoptosis. Fourth point, protection from apoptosis. So far I was saying that cancer cells are not able to undergo apoptosis. So how does it happens? What is an apoptosis? Apoptosis is a mechanism of cell death where there is a balance between pro-apoptotic and anti-apoptotic factors. Right? So pro-apoptotic factors means what? That promote apoptosis. Anti-apoptotic means what? That inhibit apoptosis. Right? So what happens? This expression of anti-apoptotic proteins will be higher in case of cancerous cell. Compared to low apoptotic pro-apoptotic factors will be lower. So it is because of the higher expression of this anti-apoptotic proteins, cancer cells becomes protected from apoptosis. And these are some examples of anti-apoptotic proteins like BCL2, BCL express. So these all proteins will undergo overexpression. So when they are overexpressed, this inhibits apoptosis. Okay. And the last one is with respect to glycolysis. What is glycolysis? Telomerase is related to end replication problem, Anit. What is glycolysis? Glycolysis is a metabolic pathway. Metabolic pathway that is involved in glucose breakdown. Right? So, the cancer cells will have inadequate blood supply. Right? So, if the blood supply is less, what will be the consequence of that? Anyone can answer? If there is less blood supply, what will be the consequence of that? If our blood flow is reduced, the rate of blood flow is reduced, what will be the consequence? The consequence will be, it will be like oxygen will not be able to travel, right? Because oxygen travels from where? Travels via blood only to different parts of our body. So when there is inadequate blood supply, obviously the person will be suffering from hypoxia condition. Hypoxia condition means low levels of oxygen. Okay. So at that situation, what happens? The cells will be expressing a kind of transcription factor which is responsible to undergo transcription and protein production known as HIF, hypoxia inducible factor. Now, what does this hypoxia inducible factor will do? This will help in production of blood vessels. So, when blood vessels are produced, oxygen supply will be restored. So there won't be shortage of oxygen for the cancer cells. So it not only supplies oxygen, but angiogenesis also supports metastasis and migration of cancer cells to different parts of the body. Okay. Next, what we are going to look is the events that occur in metastasis. So we are going to give you an example of how a colon carcinoma is developed or how a colon cancer occurs. In the first picture, you can see we are having basal lamina, right? Basal lamina is there. Basal lamina where the epithelial cells will be resting. As you can see, you have epithelial cells over there below to that basal lamina. And basal lamina is composed of what? Composed of blood vessels, connective tissue, lymphatic system, etc. Initially, you will find that we are having single proliferative cell. Slowly, gradually, the population increases, right? The proliferative cell population will be increased. After that, it has attained a stage where you call it as a small adenoma. Small adenoma means it is a kind of tumor which you cannot say that this is a cancer. It is just formation of a single mass of cells it is forming. After that, what happens? Intermediate adenoma will be there. Some more mutations, but still it is restricted to that particular location only. That's why we are calling it as an adenoma. Then you will find that it is getting converted into large adenoma plus carcinoma. Now, why did I mention carcinoma here? Can anyone say why did I mention large adenoma plus carcinoma here? The reason is that the cells have started invading the basal lamina, and it is trying to enter into the blood vessels as well as the lymphatic system, as you can see in this picture. So now this leads to what? Colon cancer. I hope so the events are clear to all of you. The events that took place in metastasis, that is colon cancer. Now, next topic under cancer is what are the types of genes that causes cancer? Any idea? Any idea what are the genes responsible for causing cancer? Cancer causing genes. What are the examples? We have got three major categories of that. If anyone would like to answer? No? No issues. I will say you. We have got three major categories of genes that are responsible for causing cancer. What are they? Proto-Oncogenes, Tumor Suppressor genes, DNA repair enzymes. What are proto-oncogenes? I guess many of you might have heard of oncogenes, right? Oncogenes are what? Cancer-causing genes. So these are derived from where? Derived from proto-oncogenes. Now I'll come into details about all the three genes individually. Tumor suppressor genes means what? It is suppressing the tumor. So we have got a category of genes that is naturally present in our body to suppress the tumor. Then we are having DNA repair enzymes. Meaning, as I said you, if we have any damage within our DNA, at that time repair machineries will be recruited. So if this repair machinery itself is damaged, then what you can expect? Cancer. So these are the three major categories of genes that cause cancer. So what are proto-oncogenes? Proto-oncogenes are normal genes whose products are responsible to undergo cell proliferation, meaning controlled way of cell proliferation, not abnormal proliferation, right? And the mutated or the overexpressed version of proto-oncogenes will give rise to oncogenes. And the proteins which is generated from oncogenes, you call it as oncoproteins. I hope so the word is clear to all of you. Proto-oncogenes are needed for normal growth and development. If any mutation occurs over here, this leads to the production of oncogenes. The byproduct of this genes is known as oncoproteins. You can see here. Majority of these proto-oncogenes participate in signaling pathways. For example, they can encode for any growth factors, growth factor receptors, signal transducers, transcription factors. Any of these components can act as a proto-oncogenes. But when this undergoes mutation, you get oncogene, and this oncogenes upon translation will give rise to oncoproteins. So, in order to give you more insight regarding proto-oncogenes, let me give you some examples. First example is RAS. Now, RAS is a protein which you normally find under signal transduction pathways, meaning signaling pathways. You might have heard about the term. So, basically, this RAS protein comes there. And it acts as an on and off switches. Now, why did I said that it acts as an on and off switches? Because it exists in RAS GDP form and RAS GTP form. This is the inactive version. This is the active version. So, depending upon the situation, it will convert to GTP or it can convert to GDP. And since it is interconvertible, you call it as on and off switches. So normally what is the function of RAS gene? Is to promote cell growth. So just imagine if this RAS got mutated, what happens? Definitely uncontrolled growth promoting signal will be brewing. See, normally RAS GTP will be there, proliferation, migration. After that, it will be stopped because it gets converted to GDP. But in this case, you can see the highlighted version of RAS GTP. That means what? Here, GDP gets converted to RAS GTP permanently. So what happens? Continuous cell growth, division, proliferation, everything. So this leads to what? Cancer. In majority of the cancer, you will find RAS gene getting mutated. For example, pancreatic cancer, lung cancer, colon, thyroid. In all these cases, you will find that RAS, one of the gene that gets mutated is RAS. Now let's look into the second example. Second example is HER2. What is HER2? Now this is HER2 gene codes for a receptor that is involved in the growth and division of breast cells. And in majority of the people who suffer from breast cancer, they have this HER2 gene being amplified. Amplified means what? Increase in number. The HER2 gene will be produced in excess. And it is for that reason such patients are said as HER2 positive breast cancer. So, third example. Third example of proto-oncogene is Mick Gene. Normally, Mick Gene is responsible for transcription of certain RNAs. Okay, they are considered to be the regulator of RNAs. Normally, I am saying. But this Mick gene is found to be associated with a cancer known as Burkitt lymphoma. Now, what is the reason of Burkitt lymphoma? Chromosomal translocation. Now, what is chromosomal translocation? A segment of DNA will be migrated or translocated to another region. As you can see, this much portion will be removed and gets attached over this chromosome. That means from 8, chromosome number 8, the segment is translocated to chromosome number 14. So this leads to what? Abnormal gene expression. So again, this leads to cancer. Next, what we are having? Cyclin D. Now, what is Cyclin D? As you all know, Cyclin D is an important protein being expressed in cell cycle, cell division phase. What is its main job? Its main job is to inactivate a tumor suppressor gene known as RB. RB stands for what? Retinoblastoma. So when this cyclin D gets mutated, what happens? It leads to uncontrolled cell growth. And these mutations are found to be associated with pancreatic cancer, lung cancer, breast cancer, as well as melanoma. What is melanoma? What is melanoma? Any idea? Melanoma is related to what? Melanocytes. Where do we find melanocytes? Skin cancer, right? So this comes under that category. So as you can see, this is our cell cycle. And I guess you all might be aware of cell cycle having 4 different phases. G1 phase, S phase, G2 and M phase. And every phase is having its own set of proteins incorporated. So I was speaking about the mutation of cyclin D, right? So normally your RB gene, if it is active, cell cycle will be blocked. But when the cyclin CDKs gets activated, so this is what? This is nothing but a kinase. What is the function of kinase? To add phosphate to the target. So who is the target over here? RB gene. So normally what happens? Cyclin CDK once get activated, they will phosphorylate the RB gene. RB gene becomes inactive. That way, cell cycle will be proceeding. Right. But what did I said you? If cyclin D gets mutated, then it leads to cancer. How is it related? See, if cyclin D is mutated, what happens? There will be continuous phosphorylation of RB gene. That means you call it as a hyperphosphorylation, which leads to what? Continuous cell cycle progression. Continuous cell cycle progression means uncontrolled cell proliferation and division. So this is how cyclin D is related to cancer. Right. Next, what we are going to discuss. So these are just 4 examples I said you. Of course, more than that is available. But since due to lack of time, just took some important proto-oncogenes and explained you. Similarly, tumor suppressor genes are there. Tumor suppressor means, as I said you already, that suppresses the tumor. So, any alterations comes in the tumor suppressor gene, it leads to cancer. So, we have 2 major categories of tumor suppressor genes. Caretaker genes, gatekeeper genes. Caretaker genes are the same DNA repair genes. They belong to the same category. See, they are responsible for maintaining the integrity of the genome. How? By repairing the DNA damage. Gatekeeper genes means what? What is the role of a gatekeeper? What does our security guard will do? It will secure us. It will provide protection to us. So like that, gatekeeper genes also provide protection. How? By inhibiting the proliferation and promoting the death of those cells which has already underwent damage. Right? So 2 categories. So if you see some examples of this one, gatekeeper genes include P53 gene, RB1 gene and APC. I guess many of you might have heard of P53 and RB1. APC maybe you may not have heard, so you can remember 2. 2 examples will be more than enough. P53 gene is acting as a transcription factor, and not only that, if any damage comes to our DNA, your P53 will be acting as a sensor for that. Sensor of any kind of damage. Right? RB1 gene, they are considered to be the transcriptional regulator. Just now we have seen in case of cell cycle. Right? And coming to the caretaker genes. I hope so many of you might have heard about BRCA1 and BRCA2. Normal function is it carries out DNA repair. If any mutation occurs, it leads to what? Breast and ovarian cancer. If it is BRCA2, it may lead to breast cancer both in males and females. Okay. Then this may not be heard by you, but this is an example of DNA repair genes. And we have got several categories of DNA repair. One category is mismatch repair. So when you go for very higher studies and all, when you study molecular biology, there you will come to know about the types of repair mechanism. And mismatch repair is one of them. And this leads to what? Colorectal cancer. So it is related to colon cancer. Okay. Now we will look into some examples of this gatekeeper and caretaker genes. So if you look into the normal function of P53, if we have a DNA damage, if we have any abnormalities in the cell cycle, or if we are facing any hypoxia condition, I said you P53 acts as a sensor at that time. So it is having 2 alternatives. Either it will induce that segment of cell or that cell into apoptosis, or it will cause cell cycle arrest. In case if the repair is damage, if the damage is being repaired, your cell cycle will be restart. Suppose if it is irreparable, in that case, it will induce the cell to undergo apoptosis. Okay, that is the normal case of P53. What happens if we have mutated P53? Whatever be the conditions, our P53 is mutated. So what happens? Instead of undergoing any of these changes, it will simply continue cell cycle. And cell cycle progression leads to what? Cancer. Right? So this is one example of your gatekeeper gene. Another example is your RB deletion. RB deletion causes what? Retinoblastoma. What is retinoblastoma? Retinoblastoma is the cancer or the tumor that occurs in eyes. So in this retinoblastoma, what happened? This particular segment gets deleted. So when the segment is deleted, you can see over here, the area that codes for RB gene will not be there. So the person will be suffering from retinoblastoma. So this is a kind of deletion mutation. Next, what you have example of how BRCA1 and BRCA2 will be acting. That is your caretaker genes example. This is our double strand. DNA is double stranded. So this is our double stranded DNA. This DNA underwent damage, right? So BRCA1 and BRCA2, what is their normal function? To undergo repair. So it will undergo repair, things will be okay. But if this tumor suppressor genes are being mutated, or if it is not working properly, then what happens? Repair will not be possible. So at that stage, your P53 will get activated. So P53, when it will get activated? Only by phosphorylation. So there will be some protein which will add phosphate to P53, thereby activating it. So once P53 is activated, as I said you in the before slide, you have got two fates. Either it will undergo cell cycle arrest or it will undergo apoptosis. So these are the genes which promotes the corresponding pathways. If we have to undergo cell cycle arrest, there will be increased in the synthesis of P21 gene, which causes the arrest. If we want to undergo apoptosis, they will increase the synthesis of BAX. BAX is an example of pro-apoptotic genes. That means that is responsible for promoting apoptosis. So the cell will undergo apoptosis. Right. And here you can see MDM2 is again, normally when the cell is normal, everything is going fine, P53 is being inhibited and MDM2 is considered to be the inhibitor of P53. So when this P53 gets phosphorylated, it will be separated from MDM2. And this leads to what? Corresponding any of this process. Okay. I hope so all the three pathways are clear to all of you regarding DNA repair genes, P53 as well as RB deletion. I hope so this is clear to all of you. Then the last category of that genes responsible for causing cancer is your DNA repair genes. DNA repair genes are what? They are involved in fixing the damaged DNA, as I said you already. So whatever any mutations has occurred, whether it is duplication, deletion, anything that has occurred, they are responsible for repairing those processes. In case if these processes are not repaired, you call it as cancer. Now, accommodating all the three genes together, I have a table for you, which will be easy for you to remember the things. First one, as I said, Proto-Oncogenes. What is their normal function? That helps in cell survival and proliferation. Now, when it undergoes mutation, it becomes dominant. That is, it becomes oncogene. And what kind of mutations you can expect in proto-oncogenes? It could be a point mutation, that is single nucleotide removal, or chromosomal translocation, that is relocating to different sites, amplification, that is increase in the number of proteins. So any of this could be the reason behind the mutation. And this mutation is considered to be dominant because you know that every gene is composed of what? Two alleles, right? And if one of this allele is mutated, this will become cancerous. That's why you are calling it as dominant mutation. And you are calling it as a gain of function mutation because here you are gaining extra function. right? It is not losing the function, rather it is gaining the function over here. Gaining the function of what? Uncontrolled proliferation. That's why you call it as a gain of function mutation and dominant mutation. Now, when you look into tumor suppressor genes, what is their normal function? To inhibit the cell survival and proliferation unnecessarily. So, survival and proliferation will be occurring when it is needed. Otherwise, it will be halted. And this kind of mutation arises by what? Either deletion, point mutation, or methylation. And in this kind of mutation, you call it as a recessive mutation. Why do you call it as a recessive mutation? Same reason. You got two alleles for single gene. If one is mutated in this case, you are calling it as dominant. If both the alleles are mutated, then you are calling it as a recessive. Because for a tumor suppressor gene to get converted into an oncogene, you need mutation of two alleles. But for the conversion of proto-oncogenes to oncogenes, you need mutation of a single allele. That's why this is dominant, this is recessive. And since it is losing the function, what is the normal function? To inhibit unnecessary growth and survival. Since it is losing the function, you are calling it as loss of function mutation. Okay. Next, what you are having, the last category, genome maintenance genes. That is, as I already said, which is responsible for repair and prevent any type of damage. This is also coming under the category of loss of function and recessive. They also arise by same pattern, either deletion, point mutation, methylation. So now you understood what is a cancer is, what are the properties of cancer, and what are the genes that are responsible for causing the cancer. Now we are going to identify the carcinogens, that means the tumor promoters. And these carcinogenic agents are broadly classified into three major categories. What are those categories? We are having radiation, we are having chemicals, and we are having biological agents. So can anyone specify me what kind of radiations we can expect which causes cancer? What kind of radiations can cause cancer? What kind of radiations can cause cancer? Very simple, very easy and very direct. Ionizing radiations. Then when you look into electromagnetic radiations, we have got UV rays, X-rays, gamma rays. Then if you look into particulate radiation, you can say alpha, beta, neutrons, protons, all this cause cancer. Right. What was the second category? Chemical carcinogens. Any idea about chemical carcinogens? Any idea about chemical carcinogens? Who can act as a carcinogen when it comes to chemicals? Actually, the list is not less. It is available in nature in wide varieties. Large numbers we are having. For example, any of the aromatic hydrocarbons can be considered as a chemical carcinogen which cause variety of cancers. Aromatic amines can cause cancer. Nitroso compounds can cause cancer. Halogens can cause cancer. There are also availability of natural carcinogens like asbestos, aflatoxin, even metals. You might have heard arsenic, cadmium, which causes lung cancer, nickels. Then other than that, we are having anti-cancer agents which causes cancer like leukemia. So the chemicals are available in plenty of categories. So try to remember whichever is possible. because it is practically difficult to remember all and everything at one stage. But we can try remembering a few, which we can, right? So, what could be an example of biological agents? Can anyone say? Example of biological agents. Biological agents will be your viruses. You are having DNA specific viruses and RNA specific viruses, which is responsible for causing corresponding cancer. You can see hepatitis B causes liver cancer, hepatitis C also causing cancer. But what is the difference? Hepatitis C is having RNA as their genetic material, but hepatitis B is having DNA as their genetic material. Papilloma virus, herpes virus, retrovirus, all are biological agents that are responsible for causing cancer. Next, so we have done with the causes of cancer. Next, what is the last category? What are the treatments we can go for or which is available? One can go for surgery. So, surgical removal of that particular portion. One can also go for radiation, even though radiation is having side effects of degradation of nearby healthy cells, but still radiation is also carried out. Chemotherapy with the use of certain chemicals. Immunotherapy, where the patient's own immune system will be made strong enough to fight against cancer. Hormonal therapy. Hormonal therapy means they will slow or they will stop the growth of those cancer cells that uses hormone for their growth. Targeted therapy, meaning they will be provided with drugs that will be targeting that specified cells. Stem cell therapy, where we are using stem cells in order to replace the injured tissues or cells. Right? So these are the various types of treatment, you can say cancer treatment. And last but not the least, we are having some anti-cancer drugs being available, which is being regularly used for treating various types of cancers. So you can say alkylating agents, anthracyclines, antimetabolites. So more or less, always remember one thing, anti-cancer agents will be those that will be either suppressing your DNA replication process, or it will be suppressing the transcription process, or it will be suppressing the translation process. So either of these three categories will come into form when we say about anti-cancer agents. So you can have a check on that. So quickly, we are running out of time. Quickly moving on to some questions which I had brought for you. Can you name the process by which a malignant cell will spread throughout normal cells? Transformation, metastasis, invasiveness, progressiveness. Name the process by which a malignant cell spread throughout the normal cells. What could be the answer? Spread throughout the normal cells. How a cancer cells will be entering the normal cells? Obviously, the answer here will be invasiveness, because the very first thing is that the cells must undergo invasion, then only it can establish the disease. Next, arrange the following sequences of tumor development in the correct order. You have to identify the correct order from here. What is the correct order? Metastasis, progression, promotion, initiation. If you understood the cancer as a whole, it will be easy for you to answer. What is the first to last step over here? The correct answer over here is 4. I mean B. What is that? First initiation, then promotion, progression, finally leading to metastasis. Third question. Which of the following is not the example of Proto-Oncogenes? If you understood Proto-Oncogenes, tumor suppressor genes, DNA repair enzymes, you can easily click the answer. So the correct answer over here is RB gene, because RB is an example of tumor suppressor genes. Name the genes which directly inhibit the cell growth or promote the cell death. The correct answer will be obviously your gatekeeper genes, because they are the one which is acting as a security guard. And the last one, what is angiogenesis? It is nothing but a blood vessel formation, right? So before winding up, just wanted to inform you, if you have any sort of technical queries, you can drop a mail at info@biotecnika.org, or you can give a call to the toll-free number. Also, you can post your feedback into the zipmessage.com. You are most welcome with testimonials, etc. Before winding up, thank you so much for listening to me patiently till the end. So, believe in yourself, but do not tell the world what you can do. Just show them. So, always remember, you must believe in yourself instead of other words or other people. Believe in your journey, believe in your hard work. That's it. So, we will meet again tomorrow for gene therapy class. So, see you again tomorrow. Take care.